have a reasonable crowd here. So my name is Franklin West, and I'm the moderator for this session. I'm from the University of Georgia Regenerative Bioscience Center. And so today's session is about stem cells and their potential use to help people with neurological diseases, disorders, and injuries. Unfortunately, there's too many neurological diseases and disorders to cover all of them. And so what we have done is we've actually compiled a group of preeminent scholars in the field to talk about some of their work. And so this morning we're actually going to hear from 4 individuals to talk about their experience and also some of their research. And so we only have an hour, so I don't want to spend too much time talking. So I'm going to go ahead and invite Dr. Jean Loring to the podium, and she's from Scripps Research Institute. Thank you for coming, Jean.
Thanks, Frank. Okay, I'm going to talk to you today, hopefully in not very much time, about a therapy for Parkinson's disease that we've been working on for around 5 years. We're hoping to have our first patients treated in 2 years. The faces that you see on this screen are of the people who are actually going to be treated by this therapy. The idea is to use cells, make induced pluripotent stem cells from individuals who have Parkinson's disease, to turn those into dopamine neurons, which are the cells that die in Parkinson's disease, and then to transplant them back into their brains. So I'm going to try to go quickly through this, and unfortunately this is what I'm going to be doing. I'm going to talk about the idea, the progress we've made, the plans we have, and mostly I want to talk about the group that has supported this research from the very beginning. We couldn't have started this project without these people who have raised money for the first few years before we could get grant support. Okay, so I have to say this because this came up again last night. There are 2 kinds of stem cells, so you can't just talk about stem cell therapies. You have to specify whether what you're talking about is taking fat out of somebody and putting it back in their bloodstream or putting it somewhere else in their bodies. That is the kind of stem cell or stem cell therapy. They may not even be stem cells, actually, we'll find that out later. This is the kind of therapy that clinics that are unregulated by the FDA, but should be regulated by the FDA, are doing all over the country. There's more than 500 of them. And there are several lawsuits right now. There's one against a company in San Diego. It's a class action suit against one company for false advertising, for saying that the therapy that they're offering was 100% effective for all of the diseases that they listed. And the patients are saying it was 0% effective for any of the diseases that they treated. And they charge $15,000 for each therapy. Okay, so these adult stem cells that come from fat, they're called mesenchymal stem cells, or they're called adipose cells. Some of these preparations are capable of making cartilage. They certainly can make cartilage in culture. It's not clear that they can make cartilage if they're transplanted before they differentiate. They can't make nerve cells, they can't make heart, and they can't make anything else. Okay? The cells that can make everything are called pluripotent stem cells. And they were first derived as embryonic stem cells from discarded embryos. This is from humans. The first ones derived from mice were in 1981. So this field is actually older than you think. The cells were derived from 5-day-old discarded embryos from IVF clinics, and these are called embryonic stem cells. The first publication on the human variety was in 1998. But in 2007, another source of cells, the same pluripotent stem cells, was developed by Shinya Yamanaka in Japan. He won the Nobel Prize for this work, I think deservedly, in 2012. And it involves taking skin biopsies from volunteers or other cell types from volunteers, and then converting those cells using some transient expression of a few genes. These cells are called induced pluripotent stem cells, and they have the same capabilities as embryonic stem cells. So some people prefer to work with embryonic stem cells, some people prefer to work with induced pluripotent stem cells. It depends on the application, but you can use either one. They have the same capabilities. And this shows how they are made from culture of fibroblasts at the top. They're reprogrammed, and they change. It's very easy to tell when a cell has been reprogrammed because they change shape dramatically. So this is not a really difficult thing for people to discern. The cells have changed. Over on the far right bottom is the, and that is not my pointer, so just ignore it. On the far right is some markers that demonstrate that these cells are, in fact, what we say they are. They're pluripotent stem cells. This is the way that I like to show, to point out and help people remember that these cells can give rise to every cell type in the body, and I do mean every cell type in the body. The only thing holding us back is the development of methods in order to make those cells in a culture dish. So some of these are easier to make than others. The cells I'm going to talk about today are the dopamine neurons for Parkinson's disease. We're in the upper right-hand corner. But there are also methods in development for multiple sclerosis therapy, for liver disease, for muscular dystrophies, for heart disease, for pancreas, for type 1 diabetes, and for macular degeneration. And in each case, the cells that are transplanted are the cells that have been made into the cell type that dies. It's a cell replacement therapy. It's not a stem cell therapy. We don't put stem cells into people. We use the stem cells to make cell types that we put into people. This is the idea behind the Parkinson's disease therapy. Parkinson's disease is caused by the death of a specific cell type in the part of the brain called the substantia nigra. It's pretty small. It's indicated there in the figure on the left. These cells send signals to another part of the brain called the striatum, and they control all the control over movements. So when people are diagnosed with Parkinson's disease, they've already lost at least half of these cells. So what that says is that you can't regenerate these cells in the brain. If you're going to get people back to the way they were, you're going to have to replace them. Drugs that have been developed to rescue these cells or keep them from dying off are extremely useful. It would be great to be able to freeze the disease so it doesn't get any worse. But if you want to restore people to health, you have to put more cells in. So this is the idea. We have a group of patients. We have made induced pluripotent stem cells from them, and then we've developed methods for making the same dopamine neurons that are lost in their brains in a culture dish. We have a reason to believe this will work because there were some experiments done in the 1980s and 1990s using fetal tissue as a transplant to restore dopamine neurons in Parkinson's disease patients. These very courageous researchers actually did clinical trials that were supported by the NIH. And what they found with 74 Parkinson's disease patients, they took cells from fetuses that are about 6 to 8 weeks old, which are really, really small, and they dissected out an even smaller part, and they had to put several tissues from several embryos into each side of the brain. So you can imagine this is challenging. It's much more challenging in the US now because of the abortion laws. It is, however, being reproduced right now in the United Kingdom under funding from a European Union grant. So there are people right now trying to reproduce the same methods that were used in the 80s and 90s. The problem with this therapy was lack of control over the outcome. In some people, there was no change. In a remarkable population, people experienced a reduction in their Parkinson's disease symptoms, which lasted for the rest of their lives. So they were essentially restored to normal with no disease pathology. However, in about 15% of the patients, there was a phenomenon called a dyskinesia. You never want to make patients sicker than they started out. The dyskinesias caused uncontrolled movements, and so that had to be treated separately, and it was clearly because of the transplant. So the reason for that is almost certainly the fact that there were other cell types besides those dopamine neurons transplanted into the brains of these patients. And when you transplant cells that aren't supposed to be there, then you can have some unexpected effects. So interestingly, this is an example of a patient who died 24 years after he had the therapy, after he had the transplants. Over here on the left is a series of pictures that show parts of the brain containing cells making dopamine. These are fetal cells that arrived in that part of the brain 24 years ago. So as you can see, they're perfectly healthy. The cells are sending out axons and connecting with the rest of the brain, which is obviously the reason why those cells have reversed the symptoms. And over here on the right, you can see a diagram of what happens after transplantation. In the green line, you can see the reduction in the Parkinson's disease score, which is a measure of the effects of the disease. And in the circles, the black line shows the increase in dopamine in the brain that occurs over time. So these transplants don't work immediately. There is an effect immediately because of the surgery, but that wears off, and then the effect becomes greater and greater with time. Okay, but we learned from these. These studies were discontinued around 16 or 17 years ago because they couldn't control the dyskinesias. So the quality control issues we learned about was the fact that you needed to use multiple fetuses. That meant each one of them had to be dissected to this tiny little bit of brain. You had to get the right part, and you had to put multiple fetuses in each person's brain. You had to get it right. And there aren't very many people in this world who know how to do this. So the fetal tissue is variable. Some of it is more viable than others. It depends on what its history is and how long you have to keep it before you transplant it. And there was no control over what types of brain cells you were transplanting and how many you were transplanting. You couldn't predict in advance. And then there was the unpredicted adverse outcome, which is dyskinesia. And of course now, in 2016, there are complex legal and ethical issues associated with the use of aborted fetuses. So this is the idea that we have. This is to essentially do the same thing, but instead of using fetal cells, we're using dopamine neurons that are derived from the patients from their induced pluripotent stem cells. So the advantage of this is really quality control. Everything is about quality control. We can know before we put the cells into the brains what kinds of cells they are. We know whether they're the right kind of dopamine neuron. We can count how many we put into somebody's brain, so we can do it consistently from patient to patient. And we're not going to put in the neurons that are believed to cause dyskinesias. So we can avoid some of the serious issues and also have much more control over the whole concept, the whole procedure. And there are no ethical issues with using a patient's own induced pluripotent stem cells. So we don't have to worry about abortion clinics anymore. But there's another bonus, and that is that the cells match the patient, so we don't need to immunosuppress people. One thing I think we all have to keep in mind is if we put cells from one person into another person, that's going to cause rejection no matter where you put them. And so those people have to be immunosuppressed. And that also means that we can give these cells again because they won't build up an antibody reaction or an immune reaction to the original transplant. So if we need to give somebody more cells later, we should be able to do that without any problems. Okay, this is just to illustrate the kinds of quality control we've built in this system. This is not what FDA recommends. This is far more than what FDA recommends. We have both classical markers that we analyze the cells at 3 different stages, actually 4 if you count the fibroblasts. We look at them when they're iPS cells. There's an intermediate phase, which is a developmental phase that's en route to the cells becoming dopamine neurons. We look at the cells once they've become immature dopamine neurons. That takes 25 days in culture. That's not such a long time. And we also don't need that many cells. The number of cells that we need for each patient can be cultured in 1 well of a culture dish. So we don't have to expand the cells, which is important because once you start expanding cells, you start accumulating genomic mutations and epigenetic changes that are probably not desirable. We have, in addition to our classical assays, we're also developing a set of genomic diagnostics, which include looking at the gene expression signature, the epigenome, the whole epigenome of the cells, and we look at the sequence by whole genome sequencing. So at each one of these stages, we're going to make absolutely sure that those cells have not accumulated any mutations, that they're the right cell type. And then after the cells have been, we can keep them in culture longer and completely mature them in a culture dish. This would happen in the patient. And then we can do electrophysiology on them and show that they are, in fact, dopamine neurons from the right part of the brain. So we have a lot of control over this process. And this is an example of what these cells look like in culture. This is an antibody staining the cells for tyrosine hydroxylase. As you can see, there are lots of cells that are positive for this marker. Also, you can see that they haven't sent out really a lot of neurites. We want that because if we try to associate cells that are well connected with each other, they die. So we had to pick exactly the right time. Okay, so we've shown that these cells will recover, they will cause recovery in a rat model of Parkinson's disease. On the top, you can see that the top line is the control, and you can see it's flat. The reduction of those other three the other three lines shows that 3 different patients had the same amount, the same effect in this rat model. And I've also shown you here pictures from the rats, which have the 3 different patient neurons. Okay, so the progress we've made is to generate the induced pluripotent stem cells we started with 8, we now have 10 to 12. We've turned them in reliably. We've developed methods for getting the same dopamine neurons every single time. That has been done by Andreas Bratt-Leal in my lab. He's the director of this project. We had our first meeting with the FDA earlier this year. It's called a pre-IND. We plan to initiate the trial in 2018, assuming we have continuous funding. In 2016, we were awarded a grant from the National Stem Cell Foundation, which allowed us to start safety trials. And over the last 2 years, we've been awarded 3 CIRM grants, most recently one on Monday of last week, that will allow us to, from CIRM, California Institute for Regenerative Medicine, which will allow us to continue on the pathway towards FDA approval. Now I want to just mention Summit for Stem Cell, and then I'm going to close. The goal of Summit for Stem Cell was to use a patient's own cells to treat their disease. And it works because it involves the patients and the advocates in the science. The patients and advocates are in our lab learning about what we're doing. It started in July of 2010 when a physician, a neurologist, Melissa Hauser, and Sherry Gould, her nurse practitioner, came to visit me at the Scripps Research Institute and asked me whether there might be a stem cell therapy for Parkinson's disease. And we spent a short time talking about it. It was so obvious and so simple, but we didn't have funding. So in 2011, Sherry Gould took a group of people to the top of Mount Kilimanjaro, including Parkinson's patients. And this allowed us to launch our project because they raised $300,000 with that. This is why it's called Summit for Stem Cells, because there's a series of summits that these advocates and the patients achieve and have achieved over time. We've had lots more fundraisers since then. They've climbed to the base camp of Mount Everest, and they just got back from a trip to Machu Picchu, a trek to Machu Picchu. These are Parkinson's patients who are doing it. So there's a sense of community and we are also part of that community as scientists. They raised more than $2.5 million to support this project for the first 5 years, and we have thousands of donors worldwide. Keep going. So as I said, the main players here, Jennifer Robb is here, and she is the president of the foundation. She has Parkinson's disease. Sherry Gold and Melissa Hauser are the physician component. We're the research component. Andres Prat-Leal is working with both Summit and in my lab, in the stem cell lab, to develop these neurons. And now I'm going to just finish with this. This is a movie which is a little bit outdated now, but I think it gives you a really good feeling for what the people are in this group are doing. Would you play this, please? Yo, guys back in the booth. Thank you. Oh, that was not what I asked for. Okay, go all the way to the end. And you should be able to just do that clicking thing on the little arrow. And if you can't, we're not going to see the movie. Yes, there you go. Just click on there. Thank you. And there should be sound, which there isn't. Can you make sure we have sound, please? Okay, so read these people's lips. They're talking about their how they had. These are the patients. They're talking about how the therapy or how they discovered they had Parkinson's disease. No sound? You want to pause it and see if you can get sound? Thank you. You might want to turn up the sound on that computer because that might be muted, and that might be the problem. You want to try that? You want to check on my computer to see whether the sound is on? Just push that little button that has the... There you go. Still doesn't work. Okay.
A friend of mine who has been diagnosed with Parkinson's, I started checking the list.
Thanks. Sorry.
Well, it all started with a little stiffness in my left hand.
And they noticed my movement was decreasing.
Even before the tremor showed up, I was very depressed.
In the early 2000s, I started losing my sense of smell.
When I compared symptoms with a friend of mine who has been diagnosed with Parkinson's, I started checking the list and saying, I've got that too and that too.
We both cried.
Dr. Hauser is the person that told me that definitively that I had Parkinson's.
Parkinson's disease almost uniformly progresses.
What I understand about Parkinson's is there's no cure.
The neurons in my brain that produce dopamine are dying off.
When your symptoms manifest themselves, as mine did several years ago, 80% of those neurons are gone.
I control the symptoms by taking some medication several times a day. I'll need more medication as time goes down the road.
When I understood about some of the side effects, I was not very excited about that.
The best hope that I've researched and have heard about is the pluripotent stem cell procedure.
It seems like magic, but it is science. The great thing about pluripotent stem cells is that they can give rise to any cell type in the body.
Sherry Gould, my nurse practitioner.
That's it? We should probably quit that anyway. I think that captured the essence. Thank you.
Thank you, Jean. So you can see we have some truly passionate researchers working on this project. So I would like to go ahead and invite the next speaker up, Miss Emily Baker. She's actually from my laboratory, and she's going to talk about some exciting work we've been doing looking at stem cell therapies in the pig model.
Good morning. My name is Emily Baker, and I'm a research assistant in the Regenerative Bioscience Center at the University of Georgia. And the title of my talk today is Transplanted-Induced Neural Stem Cells Differentiate in the Post-Stroke Brain, Promoting Tissue Recovery in a Porcine Model. So everyone in this room probably knows someone that's been affected by stroke. Every 40 seconds, someone has a stroke, and every 4 minutes, someone dies of a stroke. Our lab chooses to focus on ischemic stroke, which accounts for 87% of all strokes, and results when a blood clot blocks the flow of blood to a certain region of the brain, resulting in the loss of that tissue. There's only one approved treatment for ischemic stroke, and that's tissue plasminogen activator, which goes in and breaks up that blood clot to restore blood flow. There's only one treatment for ischemic stroke, and on top of that, there is no regenerative therapies to replace tissue that's already been lost. Our lab chooses to focus on induced pluripotent stem cell-derived neural stem cells because we think it would be a great treatment option for ischemic stroke. Rodent studies have shown that these cells are able to modulate inflammation, promote angiogenesis, and differentiate to mature neural cell types, possibly leading to cell replacement. So the ultimate dream would be with this cell technology is to have a patient with ischemic stroke isolate adult cells from this patient and introduce reprogramming factors to induce pluripotency. Then we would differentiate those iPS cells to neural stem cells for transplantation back into the patient. This would theoretically be an autologous cell therapy and would avoid the risk of immune rejection. So the West Lab uses the pig as a model to develop these INSC treatments. Historically, studies of this nature have been developed in the rodent before moving to human clinical trials. However, over 700 drugs have gone to clinical trials for ischemic stroke, and there's only been one FDA-approved treatment. And this suggests that there may be a translational gap between the rodent and the human. And that's not entirely surprising due to the anatomic differences between the rodent and the human. The rodent brain is lissencephalic, meaning it has a smooth cortical surface and contains less than 10% white matter. On the other hand, the human brain is gyrencephalic, meaning that it has those gyri and sulci and those convolutions on the cortical surface and is greater than 60% white matter. And that's not to mention the size differences between the brain of the human and the brain of the rodent. The rodent brain is 650 times smaller than the human. We think that the pig would be a great model to bridge the gap between the rodent brain and the human brain because the pig brain is gyrencephalic and also greater than 60% white matter and only 7.5 times smaller than the human brain. So this leads us to our overarching hypothesis, which is that transplanted INSCs will produce regenerative trophic factors and undergo differentiation, leading to cellular and tissue functional recovery in a pig model of ischemic stroke. So to briefly go over our experimental design, stroke surgery was induced on day 0 through middle cerebral artery occlusion. And then 1 day post-stroke, we performed magnetic resonance imaging, or MRI. 5 days post-stroke, we came in and injected 2 injections of 6 million INSCs, or vehicle only, and there were 4 animals per treatment group. And then after injection, we did MRI 1 week post-injection, 4 weeks post-injection, and 12 weeks post-injection. And then after our last MRI time point, we collected tissues for histological analysis. So the MRI sequences that we ran were T2 FLAIR, diffusion-weighted imaging, diffusion tensor imaging, magnetic resonance spectroscopy, and perfusion-weighted imaging. We also collected cell transplantation sites to assess what the neural stem cells were doing differentiation-wise after transplant into the pig brain. We tracked our cells using the human nuclear antibody, or HNA, because we were transplanting human cells into the pig. We used the NUN antibody to assess neurons, GFAP for astrocytes, and OLIG2 for oligodendrocytes. So moving into our results, we first wanted to confirm that we had a true ischemic stroke. So here you're looking at MRI images taken 24 hours post-stroke. And so our T2 FLAIR shows us a hyper-intense region in the right cerebral hemisphere that's hyper-intense that leads to, that shows us that there is a brain lesion. DWI, the corresponding DWI images show a hypo-intense region, which is indicative of stroke. And then the corresponding ADC maps show a hyperintense area, which indicates cytotoxic edema and a true ischemic event. So next we wanted to assess whether or not our neural stem cells had an effect on brain swelling or atrophy after stroke. And so what we did was we took the ipsilateral hemisphere, or the affected hemisphere, and compared it to the contralateral or unaffected hemisphere. So the graph that you see here is comparing the ipsilateral hemisphere to the contralateral hemisphere, where 0 is baseline. So 24 hours post-stroke, we see a significant increase in the size of the ipsilateral hemisphere, indicating brain swelling. And we see this in both our treated and our non-treated pigs. 1 week post-transplant, we begin to see brain atrophy. And then 4 weeks post-transplant, there is a significant reduction in that brain atrophy in our treated, in the black bars, compared to non-treated, which is the red bar. However, by 12 weeks post-transplant, that difference became less apparent, and there was no difference between treated and non-treated pigs. So this tells us that neural stem cells do not prevent brain atrophy, but it may slow the progression of that atrophy. So next, we wanted to determine if neural stem cells led to improvement in white matter integrity, and we did this through diffusion tensor imaging by measuring fractional anisotropy. So 0 is baseline, meaning comparing to the contralateral side, and we were assessing change in FA, or fractional anisotropy. So 24 hours, 1 week, and 4 weeks post-injection, we saw no difference between treated and non-treated animals in the change in FA. However, by 12 weeks, we saw that there was a significant reduction in the change in FA between 24 hours post-stroke and 12 weeks post-transplant, which tells us that the neural stem cells may be helping improve white matter integrity after stroke. So next, we used our spectroscopy data to quantify changes in neurometabolite abundance after stroke and treatment. So here we're looking at change in NAA, which is a healthy neuron marker, and we saw that 24 hours post-stroke and 1 week post-transplant, we saw no difference between treated and non-treated groups. However, by 4 weeks post-transplant and 12 weeks post-transplant, we saw a significant reduction in that change in NAA compared to the non-treated animals, and there was a significant reduction between 24 hours post-stroke and 4 and 12 weeks post-injection. We also looked at the cellularity marker choline and found a similar trend that by 4 weeks and 12 weeks post-transplant, there was a significant reduction in that change in choline in the ipsilateral hemisphere compared to the contralateral hemisphere. We also looked at the brain energy marker creatine and found that there was no difference between treated and non-treated animals 24 hours post-stroke and 1 and 4 weeks post-transplant, but by 12 weeks post-transplant, there was a significant reduction in the change in creatine compared to non-treated animals. So we also looked at our perfusion-weighted imaging data and assessed whether neural stem cells had an effect on cerebral blood perfusion after treatment. And so here the graph has changed a little bit, and we're looking at the 100% mark, where 100% would mean that the contralateral and the ipsilateral hemispheres are equal in cerebral blood perfusion. The two parameters that we looked at were mean transit time and time to peak, which are both related to the time it takes for the blood to move through the cerebrum. And so 24 hours post-stroke and 1 and 4 weeks post-transplant, we did not see a difference between treated and non-treated animals. However, by 12 weeks post-transplant, we saw a significant reduction in both mean transit time and time to peak, which indicates that neural stem cell treatment could be leading to the blood moving through the cerebrum faster by 12 weeks post-transplant. So I mentioned earlier that we also took our transplantation sites and assessed INSC differentiation in the pig brain. And so we did that through the use of the HNA antibody. So here is our HNA antibody, and the white arrows are labeling HNA positive nuclei, which points out our transplanted cells. We co-stained this with the neuron marker NUN and found that a large portion of our transplanted cells co-expressed NUN. So this is a higher magnification image showing our HNA positive nucleus co-localized with NUN, the neuron marker, and then the merge here. We also wanted to assess if our neural stem cells were differentiating to oligodendrocytes. And so we see that a portion of our HNA positive nuclei are co-localizing with the oligodendrocyte marker OLIG2, and then we see the merge here. So we wanted to quantify the percentage of HNA positive cells that were either co-localized with NUN or OLIG2. And we found that about 75% of our transplanted HNA positive cells were also co-localized with NUN, and about 25% of our HNA positive cells were co-localized with OLIG2. It's also worth mentioning that we stained for the GFAP astrocyte marker, and due to the nature of the GFAP stain, we were not able to confidently identify GFAP positive, HNA positive cells, so we chose not to include them in the quantification. So our conclusions from this is that neural stem cell treatment promotes brain tissue recovery through improved white matter integrity, improved neurometabolite abundance, and enhanced cerebral perfusion. Additionally, our neural stem cells are able to survive long-term and differentiate predominantly into neurons. So I'd like to take a second to thank my PI, Dr. Franklin West, and the rest of the West Lab, our funding sources, and our collaborators. Thanks.
Thank you, Emily. Great talk. So next, I would like to introduce Dr. Dilip Yagaval. He's a good friend from the University of Miami Miller School of Medicine, and he's going to talk a little bit about some of his research in humans and large animal models.
Thank you, Franklin. So I thank Bernie for inviting me to talk about the work we've been doing at the University of Miami on intra-arterial approaches to delivering cells in stroke. And what I'd like to cover in the next 10 to 12 minutes is how close are we to implementing this approach in patients. These are my disclosures. I don't have any patents on any of these cells or devices. We've been very fortunate to be funded with intramural grants and Florida state grants and federal grants. And the work is based on a fantastic team that I've been fortunate to have, including the small animal team. Dr. Bowan Lin is a senior researcher, and Dr. Ami Raval, and my postdoc, Pallab Bhattacharya, and the large animal team, some of whom are here, Dr. Watanabe. And the approach we've taken is to focus on the translation of delivering cells intra-arterially so that we can quickly use those findings to bring to early clinical trials. So one of the first things that is being made clear by the previous speaker is that the need for novel therapies for ischemic stroke is quite high. We actually now have a second approved therapy just as of 2015, which is endovascular treatment, which is what I do clinically, where we go up from the groin femoral artery all the way up into the occluded artery in the brain, and remove the occluding thrombus with a very strong efficacy with now about 60% of patients achieving independence if they get reperfusion with this therapy within 6 hours. However, despite that progress, what we see is even in the most recent trials of thrombectomy, almost 80% of patients are not able to return to their job. And the 40% who are not independent clearly need novel therapies. The development, however, is great because now we are able to open arteries to get cells to where they may need to be delivered using the intra-arterial approach. So the progress that's already there may actually accelerate cell therapy, especially with intra-arterial delivery. Our cells have, from the beginning, from 2009, been the mesenchymal stem cells. And what you can see is that these spindle-shaped cells are variable in size, and they can be as large as 50 microns. And I think that's where we focused our energy in the beginning, as to see how we could deliver these cells safely intra-arterially, because if the cells are big in size, they by themselves can occlude small vessels and perhaps cause ischemia. Just to take a step back, the idea that Dr. Loring presented is very relevant to our work. We see our work as a cell-based therapy where we are not trying to replace neurons with adult stem cells or glia, but we are really looking at giving a boost with trophic mechanisms to the endogenous repair that occurs after stroke, and also neuroprotection and anti-inflammatory action from these cells. As many studies have shown, these cells actually get phagocytosed in a few weeks and are thrown out. But the trophic action is what leads to benefit. The work in this field actually has been accumulating over 15 years, where several studies have shown that giving cells intra-arterially, the adult stem cells, MSC specifically, can lead to benefit. And in fact, early clinical trials were done as early as 2000, not with MSCs, but other cells, and there has been some gap until about 2-3 years ago, where again the early clinical trials have started. What our contention is that as compared to IV delivery, which is obviously the most widely available, IA delivery, which is somewhat more invasive but much less invasive than direct transplantation, has several advantages. The biggest one is that it circumvents systemic circulation and trapping of cells in the lungs and the liver, and many more cells get into the brain, into the injured area, as compared to IV. And what you can see in data from Dr. Chop's lab in Michigan is that when you give IA cells, you have an immediate, almost immediate, 4 hours distribution of cells in the ischemic hemisphere. That's the top panel. Whereas with the intracisternal and IV delivery, it takes several days for these cells to get there. These cells are ion tagged, so they're the dark spots. And with IV and IC delivery, the distribution is also very limited at about 3 or 4 days. So the ability for targeting a large number of cells to the area is terrific for intra-arterial delivery. A similar result from Dr. Steinberg's lab in Stanford, where IA delivery led to cells immediately going to the brain, whereas IV delivery led to cells being trapped in the lungs and the liver in a stroke model. What we did in my lab is to look at what happens to these cells at 24 hours after IA delivery. And we took the slice of cortex and used a technique called 3D microscopy. Can you play the movie, please? And looked at where these cells go, and these are GFP labeled cells. And what you see is the cells are not only there in the dark areas, which is the stroke brain, but they are on the surface of the arterioles or small capillaries. And if you concentrate on some of the green dots, you can see them on the surface of the vessels and in the brain parenchyma area where the stroke has caused destruction. That is certainly very encouraging to see that at 24 hours, these cells delivered intra-arterially do get into the area that we need them to get into. And the fact that they're on the surface of the blood-brain barrier and in the parenchyma suggests that the process is most likely diapedesis that leads these cells to get into the injured area. The timing of delivery still remains to be definitively worked out, but given the mechanisms of action of neuroprotection, it makes sense to give these cells early, and there are data to show that earlier delivery lead to benefit from other labs and from our lab, and I'll show you that in a second. When we started, the biggest challenge for intra-arterial delivery was posed by a study from Hopkins that showed, from Johns Hopkins, that showed that intra-arterial delivery of 1 million cells in a stroke model actually leads to ischemia in the middle cerebral artery territory in a large number of animals. And certainly that would be a no-go for translation of this to cells. So our study contended that this challenge to intra-arterial delivery may be dose-dependent. So what we did was a dose de-escalation experiment where we decreased the dose from 1 million cells in the rat stroke model to 5 million to 500,000, 200,000. At 100,000, that problem of ischemia was completely mitigated. And so when you decrease the dose of cells given intra-arterially, they do not lead to ischemia at the maximum tolerated dose. And that, when seen histologically, shows you that even at 100,000, these cells are big and sticky, and they do form these clumps inside vessels. But when you look at the cross-section in the middle, they are both inside and outside the vessel. And the last panel shows a cell half in and half out, consistent with diapedesis. And so we then took the 100,000 dose, which is the reduced dose, which is well tolerated, and did a efficacy study in the rat model. And we compared 2 time points, giving the cells at 1 hour and then giving the cells at 24 hours. And what we found was that only the 24-hour cell group had a significant benefit in their neurodeficit score at 4 weeks as compared to the other groups, including control, and surprisingly including the 1 hour group. So when you give the cells right away at 1 hour, they actually do not lead to benefit. So there is likely a need for a few hours to go by after which the cells can get into the injured area. This infarct size is reduced, and when you look at that, where the infarct size reduction occurs, it occurs mainly in the penumbral area, again consistent with a trophic neuroprotective action. So we did the similar experiment in a canine model, just like Dr. West's lab, to overcome the translational issue and to get surrogate markers like diffusion tensor imaging, similar to Dr. West's lab. And this is a endovascular stroke model where you don't have to do a craniotomy in the large animal. You take a catheter up all the way and from the groin and block the middle cerebral artery, and then have a MRI to confirm the infarct. And we give our cells at 48 hours. And what we've seen is that the treated animals have a significant reduction in their stroke size and less atrophy. And the numbers are just getting completed with our controls to have these results more statistically presentable. But with IV delivery, we have not seen similar results. What we've also been able to do, and this is a confirmatory, this is a study from a group in Korea that had similar results using the canine model, where umbilical cord cells led to a significant reduction in infarct size in treated animals as compared to controls. The controls are below. We also looked at diffusion tensor imaging. This is the neuroassessment scale that we use in the large animal. And again, what we see is, as compared to controls, we had a trend towards better improvement in the treated animals. What we're also able to do is diffusion tensor imaging, as I mentioned, where you look at the corticospinal tract at baseline after stroke and after cell therapy. And what we see is that in a control animal after stroke, the corticospinal tract size goes down, and this is tractography. It becomes very thin in the middle panel, and then it recovers some, but not completely. But in treated animals, we had 2 animals where we had a large stroke, and they recovered after cell therapy with a significant improvement, almost back to normal in the top panel in the corticospinal tract, and close to back to normal in the lower animal with the corticospinal tract. So this is pretty a powerful surrogate marker we think that could be used in clinical trials. I'm going to finish up with showing you the first trial that I led with Sean Savitz of UT Houston, called the Recover Stroke Trial, where I forgot to mention all our lab work was with allogenic cells, but we did this study with Cytomedics, the company, and they had a IND for autologous cell therapy given intra-arterially with ALDH bright cells, which are derived from the bone marrow. These were given intra-arterially at 2 weeks, and the main goal was to demonstrate safety, and the secondary goal was to look for early efficacy. And what we had was a double-blind randomized trial, randomized 3 treated and 2 sham at 8 sites. The planned sample size was 100 patients, but we stopped at 48, not due to any safety concerns, but because the interim analysis suggested that if there was a difference, we would pick it up at that sample size. And safety endpoints were any neurologic worsening that may occur after giving these cells, and proportion of serious adverse events and death. And we also looked at the improvement in disability scales. And the patient was screened at 7 days, and then they were randomized, and then a bone marrow harvest or sham was done, and the cells were dosed within 48 hours of bone marrow harvest. They were not expanded. And they were given with a microcatheter using standard techniques in the intracranial ICA, internal carotid artery, just beyond the ophthalmic artery going to the eye. And we had a total of 29 patients in the treated arm, 19 in the sham arm, pretty good follow-up. And here are the results. There was no, the groups were well matched. There was no significant difference. There was no difference in the safety outcomes. None of the groups had any neurological worsening by giving these cells intra-arterially. And there was one tumor that was seen in the control, in the sham arm, one brain tumor, none in the treated arm. And that was a metastasis from a lung cancer. There was one adverse event that was the DVT. The DVTs were more in the treated group, 4 of them, and they were felt to be unrelated to the cell therapy. So I'm going to end there. Just in the interest of time, we did not see a difference in efficacy, and we also, thanks, Franklin. We also looked at one post hoc analysis. In 48 patients, we didn't really expect to see a big difference in efficacy, but when we looked at the 4-point change as a possible measure of worsening, we actually ended up having data on any 4-point improvements over time. And the sham arm had a 4-point improvement in the stroke scale in 30% of patients. But in the treated arm, there was about a 70% chance of a 4-point improvement. This is not a traditional stroke recovery endpoint, but still a post hoc analysis suggesting that there may be a signal of benefit with the intra-arterial cell delivery of the ALDH bright cells. But to conclude about the results of the trial, the safety objective, which was the primary objective, was met, where intra-arterial delivery of cells given at 2 weeks did not lead to any safety concerns, and there was a signal of a benefit. The primary efficacy endpoint was not seen, but with 48 patients, that would be hard to achieve. And the post hoc finding of an IHSS improvement by 4 points in the treated group suggests that there may be a signal of benefit. And so we have a good proof of long-term safety of this approach with the ALDH bright cells. And I'm going to skip these slides. We are proposing a larger trial to the NIH using an allogenic approach where the cells can be given earlier, within 24 to 36 hours, again with the aim to have a neuroprotective and anti-inflammatory action in a larger number of patients. And we would use, along with the traditional endpoints, biomarkers such as DTI to look for efficacy. So with that, I'm going to conclude. Intra-arterial delivery really is a widely available approach around the world, and it appears to be safe for delivering cells. And then several early trials have shown safety of this approach. Ours, the trial that I showed you, is the biggest trial, but there are smaller phase 1b studies that have shown that this approach is safe. And then the optimal cell type and cell timing of delivery still is being worked out, but this approach of using MSCs early on seems to be at least one way where there is likelihood of benefit, a high likelihood of benefit, and larger studies are on the way to really prove this definitively in the future. Again, I want to thank my collaborators and mentors. Dr. Josh Hare has been a major force behind my going into this field, and several others who have helped with all the work. Thank you so much.
Thank you, Dilip. So today I've failed as a moderator at some level. So I know Graham is shaking his head like, gosh, really, Frank? So we're over time, but I would like to say that right now is the break time, so snacks and etc. So if you're willing to forego the break and stay with us a little longer, we would appreciate that, of course. Our next speaker is Miss Brenda Castle, and she is actually from the Parkinson's Warriors group, so a patient advocacy group. And then we're going to open it up for some quick discussion because we know it's time is growing short. So gentlemen in the back, if you could turn on the microphones that are on the table, that would be excellent. Brenda?
Well, thank you. And I appreciate the opportunity to be here to speak. I am obviously, as you can tell, I have Parkinson's. But I'm also a warrior fighter of this disease on a daily basis. And symptoms started in 2009. By 2012, I got my diagnosis. At that point in time, my leg was dragging, my right arm was dragging, my face was frozen. On behalf of all the patients, stroke victims, anyone that has a neurological disease, this is the most fascinating research, and we have to keep fighting because 3 years ago, I was in a wheelchair 30 to 40% of the time. Exercise has got me to where I am today, but that's not the only solution. So stem cell, I truly believe in it, very passionate about it, fighting for everyone that fights on a daily basis. And I'm honored to be here with this panel, and I look forward to what is yet to come, experiments, trials, whatever it can be, because the patients, the brain is a very, very powerful, powerful tool. And exercise is a key. And if we can retrain our brains with the loss of dopamine that controls movement, there's so much more that we can do. So again, I appreciate the opportunity to be here and be on such a great panel.
If anyone has any burning questions, again, I know we're over on time, feel free to come to the microphone and we'll field any questions that you may have. Thank you.
It's for Jane. Why do you have to wait to 2018 if you have FDA approval?
I'm sorry, you're asking me?
Yes.
Well, we don't have FDA approval.
Oh, okay. I thought
right. We're on our way to the pre-IND stage, and then we'll have to do the IND enabling studies. So even though it's working really well, we do have to prove to the FDA that the cells are safe, and that's the main objective of the work we're doing right now.
Safety is still the problem, okay. Yeah.
Thank you all the speakers for the great presentations, and thank you for your advocacy. I've got a question for my good friend Jean. As you know, each morning I shave with Occam's razor, so I want you to kindly explain to us why you infer that the dyskinesias are due to other cells and aren't due to inappropriate release of dopamine in the striatum.
So I failed to show the paper that demonstrates that. There has been a paper published. It's difficult to do because there's not really a rat model of dyskinesia. But there is strong evidence that serotonergic neurons that were included are responsible for the dyskinesias. It's correlative, really, and might be causative. I think the other alternative is that you just had too many neurons, but I think that doesn't really make sense given the numbers of neurons that have survived in these patients. They're not different from the ones with dyskinesia and the ones without dyskinesia. So I don't think it's just simply a matter of being overdosed with dopamine. Otherwise, I mean, it's one of those unknowns. It's an untestable at this point.
Good question. All right, well, if there's no other questions, let's thank the panel one more time and thank you for coming.